this is part 2 of stem cells. So here we are mainly going to look at the applications of stem cells, okay, how we can, with respect to cancer only, okay. So you may be aware of few things. Stem cells have enormous potential, but we will focus only on the cancer part here, okay. So first is stem cells as therapeutic agents, okay. So here you will see a new property of mesenchymal stem cell, okay. So again, what are mesenchymal stem cells? Can you remind me? Mesenchymal stem cells are adult stem cells, and they are multipotent. Okay, so they are very useful because they have this property that they migrate towards the tumor site. So if you take mesenchymal stem cells and you put something inside, they will directly go to the tumor site. They have this property. So you can use it for enzyme product therapy and also oncolytic virus delivery. So we are very fond of oncolytic virus delivery, but oncolytic viruses are cleaned by the immune system. Oncolytic viruses are not able to go to deep tissues, deep tumors, right? They cannot go, so they can only work for like surface tumors only. So you can package oncolytic virus within mesenchymal stem cells and they will go to the site. So mesenchymal stem cells are very powerful as delivery vehicles. They naturally home to tumors and inflamed tissues, making them ideal Trojan horse carriers for anti-cancer agents. Do you guys know what is Trojan horse? Can you tell me?
So, like this term is from the computer part only, Trojan horse. It's kind of a virus.
No, where did this whole thing originated, Trojan horse? Do you know the history to know where the Trojan horse was? Can you tell me? The Greeks were involved in a war, and I don't know with whom, huh? They had a wooden horse filled with soldiers, and they left it outside the enemy's boundary line, and then they opened the door and they saw a nice wooden horse, so they took it inside, and inside were the soldiers, so they just came out and killed them, right? So that's called Trojan horse. So anytime you have to deliver a drug to the tumor site, you can pack it in a Trojan horse and then it will get inside, right? So that's where mesenchymal stem cells are very important. You can deliver cytokines, you can deliver products, converting enzyme, oncolytic virus, nanoparticles, anti-angiogenic factors. So this is very good. So why do they go to the tumor site? Tumor cells secrete signals like CXCL12 and VEGF to recruit blood vessels. So MSCs have receptors for like CXCR4, which binds these ligands, and so they go to the tumor site and they home there. So now we can load mesenchymal stem cells with a lot of different kinds of things, and they will take it to the tumor site. So it's a delivery thing, okay? So suicide gene therapy. So we talked about suicide gene therapy. Remember, we gave 5-fluorouracil, and then we give the cytosine deaminase gene. So you can put the DNA for cytosine deaminase gene. It will go to the tumor site and it will get expressed in tumor cells. And if you give 5-fluorouracil, it can go in the blood, not harm normal cells, but the tumor cells will convert you give 5-fluorocytosine, not uracil. And when you give 5-fluorocytosine, the amino group of cytosine will get deaminated by cytosine deaminase, and 5-fluorouracil will form. The structure of uracil and thymine are very similar. So it will be taken up by DNA machinery to incorporate instead of thymine, and that is toxic for the DNA. DNA damage will occur and tumor cells will die. Okay, so this is high local chemo concentration. This is a chemo drug and low systemic toxicity. So again, we are very fond of oncolytic virus. So you can deliver oncolytic viruses. So oncolytic viruses are often cleared by the patient's immune system before even they can reach the tumor. So you can hide the virus inside mesenchymal stem cells, and it will protect it from antibodies during transport. Once inside the tumor, the stem cells burst, releasing the virus, and it will kill the tumor. Okay, so oncolytic virus can be packaged in mesenchymal stem cells. Now let's look at hematopoietic stem cell therapy. So these are very useful for leukemia patients. Okay, so bone marrow regeneration after high dose chemotherapy radiation. So you can even take allogenic hematopoietic stem cells for leukemias, lymphomas, multiple myeloma patients, and then you can inject it in these patients. And as I said, usually they are taken from siblings which have similar HLA. So you have to do the matching here. And once you do the matching here, you don't have a choice of taking it from the patient and putting in the patient because they're all destroyed. So you take up from a sibling and then you give it to the patient. Okay, and that helps in bone marrow regeneration. Okay, then the next is immune engineered stem cells. So immune engineered stem cells, whenever the word engineered comes, that means you are changing the immune cells. Can you tell me some ways of changing the immune cells? CAR T therapy, CAR NK therapy, CAR M therapy. So if you take the T cells out of the patient and you engineer it, that is not stem cell. But if you take iPSCs and you make them in T cells and you put the CAR there, that is stem cell therapy. So iPSC derived NK cells and then you make the CAR there. iPSC derived T cells and then you put the CAR there. You can even make macrophages. Okay, so there are different ways. So you are taking healthy donors and you can treat. So this is stem cell therapy here. So you are collecting the blood, you collect the T cells, you make cytotoxic T cells, you reprogram them. So this is conventional T cell therapy, but here you are taking iPSC cells. Who was asking me about this during lunch? Somebody was asking, you were asking me. So you were asking me, how do you differentiate?
Question was, how can we reprogram it and then use it as an active antibody?
No, it's not the antibody thing. So before I come to the antibody, iPSC cells, they are stem cells. They can make any cells. So like you said, we have pretty good idea of what kind of factors we should use if we want to differentiate to skin cells or pancreatic cells or beta islet cells which produce insulin. So if you know all the factors, you can add those factors and you can differentiate them in those cells. Okay, and then you put back in the patient. So we can add similar signals to make T cells, B cells. If we make B cells, we can make antibodies, right? So you regenerate T cells and then you can put it back in the body. So these are CAR T cells. We already know much about CAR T cells. So if they are coming from stem cells, then it is a regenerative therapy. You are doing it. If you are isolating it from the patient or healthy thing, then it is not stem cell therapy. So taking patient's T cell and genetically reprogramming is CAR. But if you take it out, you take iPSC cells and make T cells, then it is stem cell therapy. Okay, so you can make natural and you can make NK cells also, CAR T cell, and you can also engineer stem cells. So you can make stem cells do different kinds of things. So you can take an antigen, okay? You take stem cells and you make natural killer cells out of it. And these natural killer cells, you train them to fight against certain antigen. And then you put it in the patient. So NK cells will go and find the tumor and kill it. Okay, so that is completely stem cell because you are actually making NK cells and then you are training the NK cell. So it's like you are getting a child and you train it to fight against a certain thing and then you send the person to the war. Okay, so those are immune engineered stem cells. Tissue engineering, as the name suggests, like somebody, the breast has been removed, now you can completely regenerate the breast. That is a very common thing to do these days. So what you do, you take stem cells from fat or bone marrow, then you give the scaffold, then you put the scaffold, then you give enough room for the blood vessels to go, then you give the right signals so that they differentiate into different kinds of breast cells, and then they form the whole thing. Okay, This is just regular CAR T cells. So you can do tissue engineering. So the trick is that you have to give a right scaffold, the right shape, okay, and then you give the cells, and then you give all the signals, and then you have to make sure that the blood vessels are also doing it, because it has to feed the cells. So it is a pretty complicated process, but people are doing it all the time. So you have to use cells, biomaterial scaffolds, bioactive molecules, like you were saying. These are the signals which will allow the cells to differentiate into breast tissues. Okay. You can use iPSC derived T cells. So CAR T cells is very expensive, over 400K. iPSC T cells, I actually checked the price of this because yesterday we were discussing the price. So it is estimated it would be about 100K. So 100K is still a lot cheaper than taking the T cells out of that. Okay. So iPSC cells have huge potential for cancer treatment. Imagine you can use iPSC cells from the patient and make CAR T out of that. You can make NK out of that. You can make CAR M out of that. You can make NK cells which are not CAR but trained to fight antigens. Okay, so there is a huge potential here. Okay, so iPSCs can be used for cancer treatment. So here there are few examples. So PBMCs are blood cells, peripheral monocyte blood cells, and they can differentiate into many different kinds of blood cells. So you take out those, you can specifically isolate whichever cells you want, T cells, B cells, then you enrich it, you express CAR, you give it to the same patient. So this is the same patient, no rejection. Or you can do allogenic. So you take it from healthy donor, you make different kinds of, you can isolate different kinds of cells, you can make different kinds of CARs, and then you can now give it to multiple patients. So what will you do if you are doing allogenic? How will you make sure that it is not rejected by the patient?
Will remove this
What receptor?
MHC binding receptor.
HLAs. You will remove the HLAs, and you will keep some HLA which is recognized by NK cells as self, and then you will also delete TCR, right? So then you can give it to patients. Here we are taking immune cells or skin cells, other somatic cells, we make iPSC, and we directly engineer, differentiate it, and you can make simple NK cells. T cells are CAR, but these are NK cells. NK cells are killer cells. You can make it and you can give it, okay, and that will really help. Now, as I was telling you, in Japan, iPSC cells have been used to make retina. It is approved in Japan only so far, okay. So this is pluripotent cells. These are obviously embryonic pluripotent cells. This is fetal derived cells, and they have been reprogrammed to make stem cells, retinal stem cells, or brain or spinal cord, okay, and you can make different kinds of tissues out of it. So in Japan, they replaced retina of a patient using stem cells, okay. So it has not been used for diabetes, but research is going on to, the islet cells are the ones which release insulin. So the idea is to make use pluripotent cells to make these beta cells which increase insulin production. So this is a potential use because it has not been realized as yet. Most of the things have not been realized as yet. Things are going on. Now stem cells as tools to study cancer. So organoids, avatars, and chips. Okay, so let's look at it. What is it? So do you guys know what is an organoid? So when you guys are working in the lab, and suppose you're working on cancer, you are using tissue culture dish, right? So it's two-dimensional. You are plating the cells and they are growing, right? But the tumor is three-dimensional. So these days, if you just do cell culture like that, it is not acceptable. You have to use the three-dimensional structure. So that is called organoid. So Venkatesh lab developed this very easy way to make three-dimensional spheroids kind of thing, which are very useful just by using the simple comb in gel. It is just fantastic way of doing. and they have very beautiful confocal microscopy. If you treat it with drug, it's just really beautiful work. So one student from his lab came to my lab, and she created spheroids in my lab also. So they are called organoids. Okay, so let's look at them. So usually we use 2D cell culture, so that does not mimic 3D structure. So when you make an organoid, right, then there is an oxygen gradient, because inside will not get as much oxygen as outside. But when it is 2D, everything is getting enough oxygen. So if 2D does not really mimic the tumor, which is the 3D structure, if you do mouse model, it's very expensive. Okay, and then mouse immune system is not the same as human immune system. So look at this. This is a 3D structure here. Okay, the one your student made looked a lot more beautiful. This is very ugly. The way you should look at his data, like his spheroids are extremely beautiful spheroids. Okay, so these are called organoids. These 3D structures are called organoids. So they are used for many, many different kinds of studies. And nowadays what you can do, you can obviously make organoid after the cells. But you can, the more important thing is that you can take out small tissue from the patient, which is available, because you know patients undergo surgery all the time, biopsy all the time. So these patient tissues are available, and you can make organoid out of the actual tumor cells from a patient. These are called PDX models, patient-derived models. And if you do drug screening in those compared to cells, that is more effective. If you do cancer studies in those, that's much more effective than just using 2D gels, 2D cells. Okay, so patient-derived organoids for drug response. Okay. So comparing 2D with 3D, so these are patient-derived organoids, that's PDOs. So these are flat monolayer on plastic. These are complex 3D structure like a mini tumor. And here, because they are flat, they have huge access of oxygen. They don't have to compete because they are happy flat things. Here, depending on this thing, these guys will have more oxygen inside. They will not have enough oxygen. Okay, so and if when you grow 2D gels, they change over time. If you have too many passages, they change over time. Usually 3D structures are more stable, the organoid structures. So they have high correlation with actual patient response. Something you do in cells will not work for the patient, but if you make a 3D structure, it will work for the patient. So you can do a lot of things. Organoids can be used to study as a disease model. Like we work on prostate cancer, so prostate cancer organoid will really mimic prostate cancer, right? You can use for drug discovery. If you, let's say 96 well plate and in each you have organoid, if you can do a drug response there and see how effective it is, okay? It is used for personalized medicine. You actually have the patient sample. You can look exactly what is going wrong there. And it can also be used for regenerative medicine. Okay, you can expand them. You can do a lot of stuff from organoids. Okay, so this is tissue engineering, regenerative medicine, cancer research, all of these things, drug screening. Okay, and then you can isolate the organoid. You can break open the organoid, and you can actually change the cells and then again create the organoid. Okay, so there is a lot of things there. So patient-derived cancer organoids, you do a biopsy, and once you have it, you can again create a tumor organoid. You separate the cells, you replate them, and you can create that. And then you can do quite a few things. You can take normal cells, you can edit the gene, and you can create a tumor organoid. Suppose you delete p53, it will become tumorigenesis. Tell me other ways you can take normal cells and make it tumorigenic. Any method. You overexpress aurora kinase, it will become EGFR overexpress, it will become tumorigenic. Anything else? Absolutely, overexpressed MYC becomes oncogenic. These are the questions you are going to get. Okay. All right. So Applications of patient-derived organoids, personalized and precision medicine. Just imagine you are actually taking the tumor out of that, you are creating the 3D thing, and then you are actually doing it. So one of the things, obviously, you can do DNA sequencing, you can look at RNA, you can look at proteomics, find what's wrong. But you can also do the drug response. You give the drug and see which drug is working in the 3D structure. That is something we have not covered so far. So that is, drug response prediction is very accurate in 3D models. Okay, and suppose you take out the biopsy from someone and this tumor is very heterogeneous. Now in your organoid, the whole heterogeneous population is there. So you can see what combination of drug is working, right? Okay, so it is very important in studying tumor biology and progression. It will tell you what leads to tumor initiation. Because it is 3D structure, you can even mimic the immunosuppressive environment there. Okay, and you can test your therapy that way in the immunosuppressive environment. Okay, so advanced organized systems allow co-culture with other key components of the TME, such as fibroblast, endothelial cells, immune cells, and you can create a real structure in a dish, and you can study it that way. So just imagine how powerful is this system as compared to just using pure cells, pure one cell line which is immortalized, right? These cells are not immortalized, these are actual patient samples. So they're very effective. Okay, you can actually do metastasis research. So that was organoids. Let's look at avatars. Avatar is just a word they have stolen from us. Okay, you know avatar, right? Avatar. Okay, so avatar is that you are basically creating the whole tumor in the lab, right? In the 3D form, and then you are doing drug discovery with that. That's what is meant by. So that is, you test 50 different chemotherapy drug combos and you see which one works. Okay, so instead of probability, it tells you deterministic. Okay, it is much more accurate. Then I have a friend, I was trying to remember his name, Dan something. So he studies metastasis on a chip. So without going into the details, they have this kind of chip they have made, and there is something flowing there. And that is very easy. That is a very nice way, quantitative way to measure metastasis of cells, how metastatic those cells will be. Okay, so then you can even check the drugs to stop metastasis. Okay, so small chips with microchannels light with living human cells, and it stimulates blood flow, tumor cell interaction, drug delivery flow. So you can create a whole cancer on a chip. Okay. And you can also use it for measuring metastasis. Don't go in the details there. So metastasis causes 90% of cancer deaths, yet it is hard to study in 2D. So chips can visualize cancer cells crawling into blood vessels. Makes sense? And watching cells exit vessels in very distant tissue chambers on the chip, you can see everything in live photography. Okay, study how micrometastasis sleeps for years before waking up. So what happens is that sometimes tumors go and lodges at different parts of the body, like real micrometastasis, and then they will be quiescent for a while, and then suddenly they will become active. Okay. All right. The third part, stem cells for drug discovery and screening. I have already told you about that. So like you were saying, right, tubulin inhibitors. So you take iPSC cells, you differentiate them into cancer stem cells, and then you do the screening and see how it works. And you can make it very aggressive cancer stem cells or epithelial mesenchymal stem cells, and then you will know how effective is your drug. So it can be used for high throughput screening. Organoids are used for high throughput screening. What is the meaning of high throughput screening? Can you tell me? Anyone? What is high throughput screening? We just had lunch, so you guys are tired. We all need tea, which we will get at 3. But what is high throughput screening?
You compare the whatever we have with the already available library of things, and it's large scale comparison, I guess.
Yeah, that's what it is. It is high throughput, right? So how many compounds make a high throughput library? You guys are in 2025, right? What is a high throughput screening? Like how many compounds? 100, 200?
Ma'am, it's a very huge chemical space, so we have to apply various filters to reduce the chemical space.
No. How many compounds, just keep it very simple, how many compounds will constitute a high throughput screening? Here is the answer. It can be 100 compounds, it can be 100,000 compounds, right? So we used to do it in 96 well plate, which all of you have used, probably have seen at least, right? So in 1999, and this is 1999, 26 years ago, right? I joined Novartis, and the director of that place hired a person from one of the biggest car companies, Ford or something, and he was an engineer. So he said, make a high throughput machine. So he built a high throughput machine at Novartis, and that was the first one of its own kind in the entire world. And in 99, 2000, we were screening 2 million compounds in less than 1 week. And you guys, we talk about 96 well, he had in the same area as a 96 well pit, we had more than 3,000 wells. And there were nanoliters of compounds and cells, and we would sit in our office and the data would come to our computer. And then Peter Schulz, he had many spin-off companies with these high throughput facilities. So nowadays you can imagine what high throughput screening means. So you can make organoids and you can screen billions of drugs that way. Okay, you can identify novel targets and resistance mechanism. So one of the ways, let's say you develop a new drug, right? And it is very effective, and somebody asks me, what if it develops resistance, right? So what will you do to be ready for that resistance? So what you can do, suppose your drug works very well, Vanshika. So you can take your cells or organoids, and suppose it works at 1 micromolar concentration, right? So you give 1 nanomolar concentration or 10 nanomolar concentration. So some cells will die, but some will survive. You keep them in that drug for a very long time, like 2 weeks. Slowly, whoever are surviving. Then you change from 10 to 50 nanomolar. Some cells will die, but other cells will become resistant, and then they will divide. Then you increase it to 100, and then you give them some time. So this way you are slowly increasing the drug concentration, and 3 months, 4 months, at 1 micromolar those cells will be resistant. Then you look what is different in that cell line. That will give you the preemptively, in advance you will know what kind of drug resistance your drug is likely to develop. And if you use it in organoid, it's going to be better than flat cells. So you see that you can predict cancer resistance mechanisms before it happens in patients. Okay, you can do toxicity, whether this drug is going to be toxic or not toxic. Okay, then stem cells in regenerative support during cancer therapy. So this is when you give cancer therapy, it is pretty toxic. It can lead to cosmic deformity, functional loss, like speech, movement. People who have neck cancer, their voice changes after surgery. Okay, so they can actually affect the speech. Radiation leads to a lot of fibrosis, hardened, damaged tissues. So regenerative medicine can fix all of these things. Obviously, breast replacement is one of the deformity thing, right? So core biological functions. So you can build biomaterial scaffolds for tissue repair. So first thing is that if you have to build an organ, you have to provide a support. That support should be non-toxic to human body. We don't want to put anything which is toxic. The person will die. Plus you provide the support, but once the cells, so it has to be biocompatible, and once the cells grow and everything is there, you want the support to disappear. Correct? You don't want that artificial support to be there. So they should be biodegradable. because then it should be all pure tissue based thing. So that's what it is saying. You have to give a temporary ECM like framework. ECM is extracellular matrix like framework. To maintain the slope, you have to give the right biochemical signaling, and then you have to put the stem cells to survive, proliferate, and differentiate into the tissue, the kind of tissue you need. Okay, it should be biocompatible, it should degrade after the whole structure is formed, and then it should have the same mechanical properties. So if this is stiff, it has to be stiff. If this is soft, like bone is porous, so it has to really mimic the bone structure. It cannot it cannot be an iron rod there, right? It has to have the porosity so that it can get the nutrients, the blood vessel and everything. So it's a challenging thing. Okay, so this is just telling you, you don't have to remember anything here. Like kind of polymers that are used, collagen is present in us, right? So they use collagen, all the things which are present here, synthetic polymers, decellularize. So they take the whole ECM, but the cells are there. So they get rid of the cells, but they have the ECM. So they use that as a support because that is very natural support. Okay. Okay, and then they control the pH, they control the enzymes, everything there, temperature. So this is just breast reconstruction. This is very popular, but it is super, super, super expensive. Okay, this has been done in patient. So if there is a kind of stroke or some kind of damage in the heart, they are able to patch it up. They cannot build a whole new heart, but they can patch it up. So this is called cardiomyocyte patches. So they have patched it up. Okay, they have been successful in regenerating the bone. So that's quite a bit of success. You seed MSCs to stimulate bone formation. You get BMP2 to release accelerate healing. Okay, head and neck, resecting tumors in the jaw, tongue affects breathing, eating and speaking. So they have regenerated these kind of things. So quite a bit of things after cancer therapy.
Is it thyroid bone reconstruction or?
No, tracheal, this thing, yeah. So they can do that. So high risk experimental. So this is experimental but life saving for non-resectable cases. Okay, so that was that. Let's quickly go through these two things now. Gene therapy. You guys know all about gene therapy, right? You put the gene inside and you fix the thing. So here it's a really beautiful thing which has been actually done. Okay, so if there is something wrong, you can use CRISPR-Cas9 and you can fix it. If you had a choice of doing gene therapy, can you suggest some disease where we can do gene therapy? Like some kind of sickle cell anemia. It's just amazing. I know you guys don't have this presentation. If you had, the next slide is sickle cell anemia. So this is really, 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 really amazing. Okay, so gene therapy is that a gene is wrong and you fix it and then you give it. But because it is stem cell, you are fixing it in iPSC cells. Okay, so this is sickle cell anemia. So what goes wrong? Hemoglobin has just one single mutation. So this is normal red blood cell, donut-like, and this is sickle cell. And these sickle cells, they aggregate like this, and they restrict the blood flow. So normal red blood cells, they have a half-life of 120 days, and sickle cells die 10 days, in 10 days, and it's very bad. Okay. So this has been done, actually I checked, this has been done. So patient stem cells in their bone marrow make diseased hemoglobin that make red blood cells sickle shaped. So you take the gene, you fix the thing. So you take the stem cells, you can extract it from hematopoietic stem cells, easy to get, or you can take iPSC cells, you genetically modify, you give it back to the patient, and now it is normal. And it actually works. This is not experimental, this actually works. Okay, so this is just to show you that it has huge potential. You can take skin cells, you can correct them, And what is the beauty? Every cell has the same DNA. So you can correct the DNA, differentiate, and give it to the patient. Okay? This is hematopoietic stem cells. So this is hypothetical, but you know, you can engineer CAR T NK cells, you can give it to the patient, you can take stem cells and make any kind of cell, and you can edit it, right? You might have seen some science shows, science fiction shows, where they are making superhumans, like really correcting everything. Okay? All right. So if you want to turn off the immune system, you take mesenchymal stem cells, and that take care of the immune system quietening. So if you have to regenerate something or give new therapy where you don't want immune system to be super active in those cases, so then you use mesenchymal stem cells if you have to reduce the immune response. Okay? You can take mesenchymal cells and you can make so many different kinds of cells. Okay? The last thing, what you are interested in, cancer stem cell therapy. So these are not good guys, these are bad guys. They are not originating from IPC cells also. So cancer stem cells, so remember I keep saying that you take a tumor and you irradiate it, and 99.99999% cells will die, but 1 or 2 cells will alive. And they are extremely resilient, and they are usually cancer stem cells. So that one single cell is so resilient, you cannot kill it. And that cancer stem cell can form a whole new tumor in all different parts of the body. Okay, so that is that. Here they are showing you many cancer stem cells. So these are cancer stem cells. So the rest of the tumor died. These are the persisters. Okay, and then they can form really extremely aggressive cancer. So you can, so why chemo fails? Because CSCs, they are often quiescent, so they will become very quiet, they will not divide, and if they are not dividing, chemo will not work, because chemo depends on cell division, right? It is changing, so they will become quiet, they will exit cell cycle, they will come to G0 state, like they are sleeping, so chemo drug is not going to work on that, because it depends. They are mitosis inhibited. If there is no mitosis, nothing will happen, right? So they will become pretty quiet. And then there is tumor heterogeneity. You can kill some cells, you cannot kill other cells because cells are different. Tumor growth kinetics, if somebody was saying that cold tumor versus hot tumors, if they are growing slowly, chemotherapy will not work because rapidly dividing cells die faster. Okay, when you have MYC or P50, MYC overexpression, P53 mutation, that will also be a problem, because whenever tumor suppressors are gone, oncogenes are overexpressed, they can become resistant. So there are many different things which can give rise to cancer stem cells. CSCs are cancer stem cells. So how will you target cancer stem cells? So there are many ways to target it. We know what proteins are upregulated, and you can give inhibitors of that. But because we are studying stem cell, a very nice way is that you change stem cells to differentiated cells so that they don't come back. Once they are differentiated, they don't come back. So if you differentiate it, you can use ATRA, all-trans retinoic acid, and they are differentiated, then they cannot divide and make more stem cells. They are gone. So you basically do that. Okay, this is something I added for you guys. So remember, we keep talking about autologous versus allogenic kind of things. So this is for you guys, that you know, stem cell therapy is very successful already. So I said, okay, show me the successful therapies, because most of the things are still investigational. So tell me the successful thing. So these are the things for which stem cell therapy is actually very effective. So for ALL, acute lymphoblastic leukemia, multiple myeloma, CML, relapsed lymphoma, these all are treatable by stem cell therapy. So remember I was telling you about an undergraduate who was in my lab who had leukemia. So I never asked her what kind of therapy she got. So she was undergoing therapy every 3 months, right? And I told you she wore a lot of makeup to cover it up. And then she went back to Singapore and she had some therapy and she said she's cured for life. And I really wanted to ask her, but I never asked any personal questions because I was the PI. If I asked her, she would have replied, even if she didn't want to share her things. So I didn't ask her. But now I'm thinking that she got stem cell therapy, and that's why she was cured. Because all these years I've wondered what therapy she got. So this is very successful for all these blood cancer disease survival, complete remission, long-term. It's just really, really good. So it actually cures it. So stem cell therapy can be really phenomenal. That's it. We are done.